am Roja from India, from Osmania University. Just I am going to speak upon antioxidant and anti-epileptic activity. This is a kind of disease, like it's a kind of disease where mostly found in rural part of the equatorial region, where people even doesn't visit a doctor to just know for a cure. Just they depend upon mostly upon the medicinal plants like Acalyphaindica, which is mostly found in India, but most of the Chinese as well as Indians depend upon this plant to cure the disease. It's a very basic experiments what we have done, just screening the basic metabolites present in the plant extract. As we all know, the plant-derived drugs have been a part of human evolution, healthcare from many years, even thousands of years. Even before the medicine has started, plants were the basis for the medicine, and they are used in conventional as well as they are in traditional, conventional. Even they form a basic role models for our allopathic medicine also. What not we get everything from the plant. And India is a big treasure of the medicinal plants. Plant-based drugs are mostly used in India as well as China. Very even China mostly uses many plant-based drugs. Maybe this is due to different reasons like population or something like that. But the people in India as well as China, they believe that the plant-based drugs are the more efficient ones, which can cure most of the diseases. Even sometimes they go for a cure even for the kidney disease or cancers also. They depend upon plant-based medicine. India has very rich flora and widely distributed due to the temperatures. We have a very good temperatures over there, so we have a very good flora of the plants, medicinal plants. And these herbal medicines are being used in Ayurveda, Unani, Siddha, and now these days even plant-based medicines have just evolved like anything. Now this is a plant which we have selected, which we have done some survey in the rural part of India, and we have selected the plant which looks like, and it's found everywhere in the hottest part of the India, which is called Acalyphaindica, belongs to the kingdom Planta, and it's not really classified also properly, but it's given a name Acalyphaindica for a biological name. So the plant was very new of its kind. We have to get the plant, get it authenticated. We have got the plant from Mahbubnagar district, in Andhra Pradesh, India, and we have to get it authenticated because the plant is found in very rural part of India. Leaves were used for the extract. We extracted in methanol and hexane solvents. And these solvents, after collecting the leaves, were washed thoroughly, and we should not dry them in the hot sun, so we may lose the metabolites present in them. We have shade dried them at a temperature of 38 degrees to 40 degrees. After drying, we have made the fine powder, with the few fragments and powdery part, and that was used to extract with the help of methanol and hexane. The plant extracts were screened for presence of the biological active compounds like alkaloids, carbohydrates, glycosides, saponins, proteins, phenols, tannins, reducing sugars, flavonoids, and standard phytochemical screening methods were used to just find out what metabolites are there. And as we had with the methanol and hexane, we found these compounds. Proteins were not there in methanol, whereas in hexane the protein compounds were present, and similarly with some other metabolites. So this is the same thing which gives the summary of the table. Phenols were present in methanol and hexane extract, and where proteins were absent in methanol and present in the hexane as well. So basically we have started our work with the antimicrobial activity with different microbes to know whether really the plant has some active compounds or not, and then we have conducted some experiment. The data which I am not showing, which would be very lengthy. And then we have just conducted our antioxidant activity, like which is to be must to know, really to know about the free radicals which protect the human body from diseases, and the oxidative stress, to know the oxidative stress which plays a major role in development of chronic as well as degenerative elements such as cancer, autoimmune disorders like rheumatoid arthritis, cardiovascular and neurodegenerative disease as epilepsy. So these antioxidants, they act as a free radical scavenging for preventing and just repairing the damages by which is caused by reactive oxygen species. So like with that's the reason we have made it important to the antioxidant activity, and which was done by this procedure which is called DPPH, was used to evaluate the free radical scavenging. And this was the procedure which we followed, and like we have taken the readings at 570 nanometer using UV spectrophotometer, and this was the result. As the amount of the DPPH, as the amount of the plant sample was increased by 10, 20, 30, we found that the activity also has increased. The activity of the extract has increased, the oxidative activity. And for oxidation, again we have done one more method which is called as FRAP method. Reducing power capacity was assessed with the help of potassium ferricyanide solution, and this was the procedure which we have followed. And total mixture with the time lapse and all, and finally the absorbance of the solutions were done at 700 nanometer. And here also we have got a very good result, where we have found as the amount of extract increase, the reducing power also has with a very good result which has given. Now coming to epilepsy, and this is a common and diverse set of chronic neurological disorder which is found mostly in the equatorial region. It occurs due to abnormal electric activity in the brain. Like there is sometimes people give some symptoms like seizures when it is severe. Abnormal excessive hyper synchronous neuronal activity also occurs in the brain due to epilepsy. And about 50 million people worldwide are suffering from epilepsy, and 90% of epilepsy is just in the developing countries. And when the seizures occurs, there are two types. One is partial and generalized seizures, and these when seizures occur, I don't want to get into details of seizures. There are two types, but seizures induce damage in the hippocampus neurons. Once there is really damage in the brain, I think it's very difficult to manage with the brain neurons. And the calpone-3 may be deregulated in the patients. It's still not much research done in this kind of disease, but we found that calpone-3 was getting deregulated when the disease occurs. And finally, what we have done is we started our experiments with rats. We have divided them into 5 groups, each group having 6. For the first one was a normal control, and second one was the group with which was a sham control, which was just injected with the saline. And the third group, FECL group, received intracortical injection of freshly prepared FECL solution, like as the values are given. And and fourth group was after FECL injection, we have started injecting 20 mg per kg the Acalyphaindica plant extract. And the fifth group was there was a drug which is used by the allopathicians like diazepam, which is called. We started just giving to see how far the plant has the activity of controlling the disease. And we have just counted the wet dog shakes. Once the disease is induced, the mice starts moving its head in the radial direction, and we started counting the number of wet dog shakes once the epilepsy was induced for the rats. And these were the healthy rats. The diseased rats, we could not take the video and I could not get it, but these were the healthy rats which were just divided and put into separate chambers. And see, the iron-induced experimental epilepsy model was prepared by injecting 8 microliter freshly prepared aqueous solutions containing 10 millimolar FeCl2 over a period of 5 minutes in the sensomotor cortex by using this stereotaxic apparatus, which is commercially available. And the behavior observations were noted once the FeCl was induced after 4 hours of induction, and animals were placed in individual observation chambers, and these wet dog shakes were just counted for at least for 1 hour continuously to know like how many shakes we observe per hour, and were identified as rapid rhythmic shaking of head in the radial motions, and signs were counted and presented as number of events per hour. And administration of FeCl, as I told you, it resulted in that, and AI, what we are showing in the result, indicates Acalyphaindica, and the DZ is diazepam. And this is how like significant, we after inducing the Acalyphaindica, we have just observed significant decrease in the wet dog shakes, and we didn't observe any seizures for the rats once after induction of epilepsy with FeCl2. And this was the final result what we have observed, like only the rats, the third group of rats which we induced and left without any treatment, they had they have given very more shakes, which I will show in the next slide. And these were the rats. Like our plant sample has given more effective, it was more effective than the diazepam in the epilepsy induced rats. And these were the healthy rats after treatment of the treatment with the plant sample. And this was after 7 days of the treatment. We have taken the like wet dog shakes, like which were 300 for the FeCl induced, and it was only 100 for our plant sample induced plant sample given rats, and for the FeCl it was 110. Means our plant sample has worked more effectively than any other. Now coming to conclusions, we can say that the methanolic extract, this is a very basic experiment, but still we can say that methanolic extract was more effective compared to the hexanic extract, like which worked against the epileptic. And in this present study we can say that really the plant sample is very effective, and we are into further studies with NMR and many more metabolic studies with the rats and so on. Thank you. Any questions? Yeah. We don't know any reason. Maybe that is due to, it's mostly the equatorial region. Maybe due to heat conditions. In India, as far as my observation, it's due to like hottest places they get. In summer mainly they get the seizures. Don't know, like we exactly don't have. Yeah. We have to. Yeah, but comparatively, as far as I have observed, we have very less literature on this. Like as far as my observation, I found like mostly it is in the equatorial region. Is that 90%? Means the disease prevalence, 90% of the patients are in the those places. What I mean to say.
What made you choose this plant for the first place?
Actually, we have visited some villages around in India, like where the disease is more prevalent. Like there was an observation, like when there was some statistical analysis about the diseases. So we have just seen that in one part of India, where the people with epilepsy were more. And we have found them not even going to a doctor, they were just taking the plant. They had some local what you call some doctor which the people believe in rural places, they were using this plant. So then we have done some microbial experiments, then we have started.
Do you use like brain activity measurements?
No, we have to do it. Like we are into that. We have to start.
So that would be a more
Yeah. Even I have the plan. Yeah, even I have. Yeah. Yes, I want to do. Like we are planning. We have started.
yeah, some sort of electrical activity.
Yeah. Even we have just known that there's an instrument and we have to do that. I am on the